Let's dive right into this. We are about to master the very foundation of biological life. Think of the cell cycle not just as some dry biological process, but as a highly regulated, beautifully cinematic factory assembly line. From the exact moment a cell is born to the split second it divides into two identical daughter cells, it's on a rigorous journey. And mastering this baseline physiology, it's absolutely crucial. It is the key to understanding literally everything else that follows in pathology and pharmacology. But before we look at how this factory runs, we gotta ask the most important clinical question. What happens when the factory's security actually fails? Why do we care so much about this? Well, when this perfectly timed assembly line breaks down, when it accidentally passes along defective, damaged products without stopping them, that leads to uncontrolled division. And that, my friends, is the root cause of cancer. Understanding the healthy cycle is your roadmap to understanding exactly how tumors form. So here is our factory blueprint roadmap for today's explainer. 1. Interphase and growth. 2. The highly debated S phase. 3. The strict security checkpoints. And 4. Culminating in mitosis, the big split. All right, let's kick things off with Section 1, Interphase and the resting zone. Now, interphase is by far the longest part of the cell's life. But you might be wondering, what about cells that just don't want to divide? Like what about your cardiac myocytes or your neurons? Well, that brings us to the G0 phase. I absolutely love thinking of G0 as a cellular vacation. It's a non-dividing state completely outside the active cycle. The cells are very much alive, they're doing their daily jobs like resting hepatocytes in your liver, but they are entirely taking a break from preparing to divide. And for neurons, this vacation is actually often permanent. Now, for the cells that do get the green light, we track the chronological progression of the factory floor through interphase. First up, we have the G1 phase. The G stands for gap, or growth. Here, the cell is just carrying out regular business, synthesizing proteins, and growing. Then we hit the S phase, which we're going to detail next. And finally, the G2 phase. By the end of G2, the cell looks like a massive balloon of cytoplasm and duplicated organelles, all prepped and waiting for the ultimate division. Let's move right along to Section 2, S phase demystified. As we set the stage for the highly critical unzipping and replication of DNA, we hit a classic high-yield wall. This is a standard doubt that trips up almost everyone at some point. The question is, S phase ke baad ploidy change hoti hai kya? Do we become 4N? Seriously, so many people get this wrong. Notice how clearly we can split this concept. First, let's focus on the actual amount of genetic material. During S phase, the DNA absolutely doubles. We go from 2C to 4C because every single thin string of DNA, the chromatid, is copied and pasted. But contrast that with the chromosome number. Human somatic cells have 46 chromosomes. Because those two identical chromatids remain pinched and joined together at the center, the centromere, they still only count as one single chromosome. So the absolute crucial takeaway here is, the chromatid mass doubles, but the chromosome count strictly stays at 46. Ploidy remains 2N, no exceptions. Moving on to Section 3, checkpoints and the role of P53, essentially our airport security. You really can't run a massive cellular factory without strict quality control. Throughout this cycle, specifically at the G1/S boundary and the G2/M boundary, we have checkpoints, and this brilliantly illustrates their function. Checkpoints are like strict airport security scanners. Agar DNA में कोई damage है, security luggage block कर देगी. If there's a mutation, a miscopied protein, or literally any DNA damage at all, security steps in and entirely blocks the luggage from getting onto the plane. The chief security officer at these checkpoints is a tumor suppressor protein called P53. It is literally known as the guardian of the genome. If P53 detects DNA damage at, say, the G1 checkpoint, it blows the whistle and halts the cell's cycle right there so repair mechanisms can try to fix the problem. But what if the damage is too severe? What if it's completely unfixable? Well, P53 triggers apoptosis, which is programmed cellular self-destruction. It literally sacrifices the cell to save the whole body. So what happens when the guardian falls asleep on the job? If the P53 gene itself is mutated, such as Inley-Frommeny syndrome, that airport security scanner is essentially turned off, unplugged, damaged. Defective cells easily bypass security and just keep dividing, copying their errors over and over again. This is exactly how uncontrolled tumor growth begins. It all traces back to a failure in these microscopic factory checkpoints. Finally, we reach Section 4, mitosis, the big split, where all this preparation pays off. Mitosis is the actual division of the replicated DNA into two distinct nuclei. You can remember the order of operations with a really simple mnemonic. Please make another two cells, or PMAT. First is prophase, where the messy DNA condenses into visible, tight little X shapes. Then metaphase, where they all beautifully align right in the middle of the cell. Next is anaphase, where they are violently pulled apart. And finally, telophase, where new nuclear membranes form around the two new sets of DNA, right before the cell membrane pinches them into two whole new cells. But let's pause the timeline right at anaphase. Here's another major conceptual hurdle that trips people up. What exactly is the difference between chromosomes and sister chromatids at this specific moment? Because remember, earlier we said copying them in S phase didn't change the chromosome number, so what gives? Okay, here is the secret. The anaphase promotion. During metaphase, those identical sister chromatids are still glued together at the centromere, so they still count as one chromosome. But in anaphase, organelles send out spindle fibers that grab onto them like a brutal game of tug of war. The exact literal fraction of a second those sister chromatids snap apart and separate, boom, they are promoted. They are officially counted as individual independent chromosomes heading to opposite poles. Let's distill this entire factory journey down to the absolute highest yield takeaways you need to remember. First, G0 is your cellular vacation, your resting state. Second, in the S phase, the DNA amount goes from 2C to 4C, but ploidy stays strictly 2N. Remember that. Third, P53 is your genome's rigorous airport security scanner, preventing tumors. And finally, anaphase is the precise moment chromatids are promoted into individual chromosomes. We've seen how perfect this biological machinery is, but I want to leave you with a thought to bridge this into the real world of medicine. If a powerful chemotherapy drug is designed to specifically target and block the S phase, what exactly happens to our factory's assembly line? And why would that be the ultimate weapon against cancer? Think about what we just covered today, and I'll let you ponder that. Keep learning, keep questioning, and I'll catch you in the next explainer.